Sorry, I'm trying to look as happy as I can for this video. But in this video, we're going to discuss secondary active transport. That's why we're all smiles here, baby, all smiles. But in the previous video, we discussed primary active transport. Now we're going to discuss secondary active transport, which obviously is another example of or another form of active transport across the cell membrane. So in this example, or in secondary active transport, our example we're going to use is glucose moving from a low concentration to a high concentration. So that's where the active transport, it requires ATP. But pretty cool with the secondary active transport. There's a little unique quirk or an additive, if you will, to our primary active transport. So the sodium, in this case, actually moves from a high concentration to a low concentration into the cell. So the glucose, the substance being actively transported, moves from a low concentration to a high concentration into the cell. Guess what? Sodium also moves from a high concentration, or it moves from a high concentration to a low concentration into the cell. So let's break this down. So the carrier, in this specific instance here, we're just using an example of glucose absorption in the intestinal tract. There's a multitude of other secondary active transports within the body, but this is a real-life example to kind of paint the picture, if you will. We'll also mention and delve into an analogy at the end, but let's get into the brunt of get through the kind of main portion of the information, then we'll get to that. But in the glucose and sodium examples, the carrier, this specific carrier into the cell has a binding site for both glucose and sodium. So sodium, it has the correct specificity, so it binds, no problem. Has a strong affinity, and this sodium binding actually makes the carrier specific to the glucose. So before the sodium binds, glucose, not specific to the carrier. Or the carrier's, not specific to the glucose. But once that sodium binds, the carrier kind of transforms to being now specific for the glucose. So once the sodium binds, boom, glucose can now bind because there's correct specificity of the carrier. And then the sodium and glucose are transported into the cell at the same time. So in this instance, the co-transporter, it's called, is the sodium. So a co-transporter is required to make a carrier specific for the substance being actively transported. So without the sodium, glucose isn't moving into the cell. So the co-transporter always moves from a high concentration to a low concentration, where the substance being actively transported moves from a low concentration to a high concentration. In this case, sodium moves from a high concentration to a low concentration into the cell. Glucose moves from a low concentration to a high concentration into the cell. But that glucose is reliant on the sodium to bring it into the cell. That sodium is the co-transporter, which is required to make the carrier specific to the substance being actively transported. In this case, it would be glucose. So ATP is honestly required to set up the concentration gradient of the co-transporter here. So what that means is sodium moves from a high concentration to a low concentration into the cell, allowing the glucose, or bringing the glucose with it, essentially there. But guess what? That means sodium is now in a higher concentration, or not as high a concentration out of the cell compared to the inside of the cell, because there's more sodium in the cell. So we need to reset that concentration gradient of sodium, since there's getting to be a higher concentration of sodium in the cell. So guess what? We have an active transport mechanism, a primary pump, primary active transport, to get this sodium out of the cell from a low concentration to a high concentration, because there's still a higher concentration of sodium out of the cell compared to the inside, but the concentration gradient is not as steep here. So how do we get this sodium from this low concentration into the cell to a higher concentration out of the cell to allow this concentration gradient to be high enough to transport the glucose in? We have the primary pump. So we just discussed it in the last video of the primary active transport. The sodium in the cell, ATP, the phosphate group phosphorylates the carrier, allows the sodium to move and be pumped out of the cell to set up that concentration gradient so it can bring more glucose into the cell and then just get pumped back out, bring more glucose into the cell, reset the concentration gradient, boom, Sodium brings the glucose from a low concentration to high concentration into the cell. Sodium gets pumped back out through primary active transport, and it's just a continuous cycle there. So the secondary active transport is dependent on primary active transport, essentially, because without that primary active transport, sodium is not going to get pumped out of the cell, which means there also won't be a high enough concentration gradient of sodium out of the cell compared to the inside of the cell, so the sodium won't be able to make the carrier specific for glucose to move from a low concentration to a high concentration. So the primary active transport is major key here, as DJ Khaled would say, major key, bro. That's a horrible TJ Khaled accent, but the ATP is required to set that concentration gradient up of the co-transporter. In this case, it's sodium, but it might be another molecule in a different secondary active transport. But think of an analogy, an example of the secondary active transport in an analogy form would be, think of like whenever a 21-year-old chases a shot of liquor with some pop. So need the pop to get the shot down the throat. So essentially, in this instance, the liquor would be the glucose moving from a low concentration to high concentration. The pop, Mountain Dew, whatever, Pepsi, would be the sodium, the co-transporter in this instant. A 21-year-old, not me, I am 21, but I'm not in this discussion here, so a random 21-year-old, Billy, the 23-year-old, takes a shot of liquor, the low concentration to high concentration, which goes from a low concentration to high concentration gradient. Boom, but you need that pop to swig it down, that co-transporter, if you will, that moves from a high concentration to a low concentration to allow that glucose or shot of liquor to go down the throat. So that shot of liquor is dependent on that co-transporter to be moved down the throat. So in this situation, obviously, shot of liquor, it gets chased with the pop to allow it to go down the throat, but with secondary active transport, glucose moves from a low concentration to a high concentration, but it needs that co-transporter to set up or to make the carrier specific for that substance being actively transported. And then obviously that ATP is required to set up the concentration gradient of the co-transporter to move that sodium back out of the cell to make it a steeper concentration gradient, so it can keep bringing more glucose from that low concentration to a high concentration gradient. So secondary active transport, that substance being actively transported moves from low concentration to high concentration gradient, but the co-transporter is required to move from a high concentration to a high concentration gradient, to a low concentration to move or to make the carrier specific to the substance being actively transported there. So the substance being actively transported, low to high, co-transporter high to low, ATP required to set up that concentration gradient, and the secondary active transport is dependent on primary active transport to pump the sodium, if you will, back out of the cell to just do it all over again, the continuous cycle of life, and that's why we love secondary active transport. Oh, I forgot to press pause.